then goes to disturb histone methylation, which impairs the activation of genes, and many of which go to the development of the hippocampus. So for my purpose, it was to study the effects of Kabuki Syndrome on learning and memory retention in both heterozygous transgenic Kabuki mice, which we call HETs, and wild-type mice, by observing their phenotype, which was their behavior, during the novel object test, and then comparing differences through the data we gathered. So we gathered our data from a test called the novel object test, which was performed in open field chambers for 5 days. Let's see. So here's a video of the test being run. And so on days 1 through 3, the mice are placed in the chambers for 5 minutes each to allow for habituation. So what habituation is, it's essentially getting the mice used to their environment so they aren't anxious, which could skew the test results in the end. On day 4, each mouse is placed back in the chamber for 15 minutes with 2 orange objects. And then on day 5, which is really the essential day, the mice are placed back in the chambers for 15 minutes, except this time, one of the orange objects has been replaced by a novel green object. And so what I did in the experiment is on day 5, the mice were video recorded, and I just watched all these videos, and I recorded how long the mice spent exploring both the novel object, which is the darker green, it's actually green one, and the familiar object, which is like the white looking one. And I did this in the button box program. So based on previous research, the wild-type mice should explore the novel object way more than they explore the familiar object, while the het mice, which are the ones that have Kabuki Syndrome, will explore the objects about the same time. And so why this is because the hippocampus is responsible for that spatial memory, which is remembering where things are and what they looked like. And so if the mice cannot do this, the het mice will not recognize that the green object is new, and so they'll just think that both of them are new, and so they'll be more likely to explore both of them the same amount of time. So when we gathered our data, we put them into 4 different graphs to gain a cohesive understanding and a more detailed view of the results. So in this first graph, it just shows the raw explore time in seconds of the mice, which is on the y-axis, for both the wild type and het mice in bins 1, 2, and 3. And so what bins 1, 2, and 3 are, we split the time that the mice were in for 15 minutes into 5-minute increments because it allows us to gather a more detailed view of our data. And so as you can see from this, there's no significant difference between the exploration in the wild type and the het mice. However, the het mice did explore more during all 3 bins,